and fellowship in oculoplastic surgery and orbital surgery. And he's an assistant professor at the Department of Ophthalmology at Stanford University. He completed his ASOPRS Oculoplastics Fellowship here in Toronto, which was a 2-year fellowship, and he was served for a chief fellow during those 2 years. After finishing his fellowship, Dr. Wu became faculty at the New York Eye and Ear Infirmary, Mount Sinai School of Medicine, where he obtained support from the National Eye Institute to develop stem cell therapy. And that's to treat corneal blindness and regenerate the surface of the eye. He currently leads the Stanford Ophthalmic Stem Cell and Regenerative Medicine Laboratory, where he focuses on the molecular pathogenesis of ocular and orbital diseases. His research focuses on using induced pluripotent and adult stem cells to regenerate the ocular surface. He has pioneered the use of corneal organoids to create transplantable ocular tissue and to model human disease and development. Additionally, his lab has developed novel techniques to reverse the hallmarks of aging in human eye cells. It is an absolute honor to have Dr. Wu with us, and without further ado, I will offer him the podium.
Great, thank you. So good morning, everyone. It's great to be back in Toronto. And I first would like to thank Dr. Curtis, Dr. Yan, Dan DeAngelis, Dr. Rai. It's really been an incredible return. And what I'm hoping to do today is really give a overview of kind of the research that's been going on in my lab, but really tell a story. They say the human brain is wired to hear stories and narratives, and that's the way we learn and remember things. And what I'll tell really is a story of my path from fellowship here to beyond and kind of where I'm at now. When you leave a place, you really learn and you come to value what you had there. And when I was here as a fellow, as chief fellow, I was able to meet a lot of the faculty in all the different divisions. And I think what you have here is very special. The critical mass of students, of fellows, of residents, and the incredible breadth and depth is really outstanding. I frankly have not found that elsewhere. So I'm overjoyed to be back. Let's start off with a few of my financial disclosures. I'm a consultant for Turn Bio, which is a regeneration startup out of Stanford. And I am on the advisory board of Accelerate, who's working on a thyroid eye disease therapy currently. Okay, so regenerative medicine, how did that all begin? Well, when I was a MD PhD student at the University of Washington, I did my PhD in signal transduction and visual transduction, so the retina. And I thought, well, this is very, very interesting. I went to Salt Lake City at Moran Eye Center, and I thought, I'm going to go into retina. Retina's great. But then I found oculoplastic surgery. I found some mentors who it clicked. But at that time, this was 15 years ago, there wasn't much in terms of research going on in oculoplastic surgery. Ray Douglas kind of started up. He was working a little bit on thyroid eye. Alon Kahana was at Michigan. He was working on zebrafish and kind of orbital development, but there wasn't much going on. And I thought, okay, well, let's see what happens. So I did my residency. I was very, very lucky to match here at Toronto. And I love the surgery. It was a big surgery for me. There were a whole range of procedures. You could do orbit, you could do lacrimal, you could do eyelid, you could do some cosmetic if you wanted. I thought, wow, this is great. I kind of like the breadth of it. And during medical school and graduate school, the whole pursuit of research can be somewhat isolating. You're in the lab, and when you're back in the clinical setting, you're working often in a team. You have your residents, your fellows, you're working on patients that are complicated, and there's immediate tangible results to the work you do. You do a surgery in an ophthalmology, we have great outcomes generally. So it can be very, very satisfying. So when I finished fellowship, I thought, am I going back to the lab? Is it worth it? Because you have such a range of opportunity. And at that time, Dr. Ostreicher, Dr. Harvey, and my mentors were very encouraging of my desire to kind of delve into the research realm. And at that time, there were incredible researchers here at Toronto. Frida Miller, Derek van der Kooi, really opening up the field for stem cell in regenerative medicine. And in 2011, the Nobel Prize was given to Shinya Yamanaka from Japan, that you could turn back the clock from an adult cell to an embryonic cell. So in many ways, we had opened up, maybe Pandora's box, but we had opened up the way to actually rejuvenate tissue and to create plentiful tissue for transplantation. And in oculoplastics, where we're dealing with large reconstructions, where the eyelid is gone, lacrimal gland is gone, sometimes the eyeball is gone, socket is all contracted, etc. Hey, having new tissue would really help people. So I thought, well, wow, I'm kind of poised right on the precipice. I go one way or the other. And luckily, I met Doug Jabs, who was then chairman at Mount Sinai. And they said, hey, we'd love to bring you in and we'll give you time to do research. I started at Mount Sinai. And again, it's very fortuitous. Just like when I was in Salt Lake City and I found a mentor in oculoplastics who kind of clicked, the incubator there was for corneal regeneration. So I thought, okay, let's work on that interface between cornea and oculoplastics. Let's think about how we can reconstruct the ocular surface. And that's really how it all began. Funding from NEI, and the rest I'm going to try to depict for you today. Oftentimes, when you approach a problem, it is kind of like when you're given lemons, can you make lemonade? If you're given a certain tool and a certain problem, if you find another tool, hey, maybe there's another problem to get into. So I think that has been a little bit of the ethos of the laboratory, is that through collaboration, meeting people, and by kind of fortuitous discovery, we think about how can those discoveries be used for ophthalmology and oculoplastics. Today it's going to be more of like a whirlwind tour of how, wow, there was a problem, we have a technology, how can we immediately get to proof of concept that we can use that technology, that new discovery in ophthalmology. So I think if you're sitting down in a room, people are giving a talk, you always think, okay, I'm an ophthalmologist, I'm an orbital surgeon, how can we apply it? So it's going to be really a whirlwind tour. Just very broadly speaking, for regenerative medicine, there are a few different techniques that we've focused on. Probably in the past 5 years, one of the biggest splashes, changes, technologies has been single-cell transcriptomics. I'll talk about that. Reprogramming was really that Nobel Prize and what can you do to a cell. Rejuvenation is an outshoot of that. Instead of going all the way back to an embryonic state, let's go back 5 years, let's go back 10 years and take time off an adult cell. And finally, if we do turn the clock back all the way to an embryonic state, can we nudge the cell, not to go back to skin or blood where we got the initial cell from, but can we turn it into ocular tissue? So the first couple of projects that I'm going to very, very briefly talk about are a few things in the lab to give people an idea of, if you do go into the lab, how can you take a problem and try to solve it and make it really translatable from the bench to the bedside. This was a work that we published in TVST that looked at the production of a hydrogel. Hydrogel is kind of like a viscoelastic, and can it be used really to stop symblepharon formation? Symblepharon, we know, a very, very tough problem. Even back in the day, it's like, okay, you take a symblepharon, you take a glass rod, you break it up. I heard stories of residents going and breaking up the symblepharon every couple of days, trying to improve the patient's blink and ocular surface. Those inevitably rescar, and we know that's not the way to do it. You kind of stir up the inflammation. But do we have good techniques to deal with it? Amniotic membrane, a few things like that. But ultimately, we still see in our clinics lots of patients with symblepharon, and you can see that eye on the right is not going to see well. Okay, that goes without saying. So what happens? Well, you have scarring of the conjunctiva. Anytime you have two epithelial surfaces that are raw, they largely will adhere. We harness that to create our permanent tarsorrhaphies. We essentially denude the surface of the margin and we create a permanent tarsorrhaphy. Well, many years people have been doing alkali injury to the surface of the cornea and causing formation of corneal opacity. But people hadn't really looked deeply into the fornix. Again, oculoplastics, people haven't delved into lab work in largely for oculoplastics. So we thought, well, anytime, if we're trying to treat symblephra and we have some potential treatments, you want to create an animal model. So there are a couple of projects where we've created animal models really as a stepping stone to testing of therapeutics, et cetera. So here we showed if you injure the fornix and the conjunctiva of a mouse, you can create symblephra. And we did that in a controlled fashion. You know how long, what kind of injury do you place? And we foreshorten the fornix. This is shown in H&E that the fornix is vastly smaller and the inflammation is apparent. And if you do it over the course of 7 days, 5 days, we followed the time course of the injury. And essentially, we were able to create symblephra. Well, we thought, okay, that is very, very useful. But what if we had something like a viscoelastic? We were collaborating with Eric Appel's lab at Stanford. He's a bioengineer, and he creates different types of viscoelastics and hydrogels that will stay onto surfaces and not go away. So these, he had tried intraperitoneal, and I thought, oh, let's try this product on the eye. And in fact, what we showed here, as you can see, that first uninjured and injured, clear corneal opacity, and there's fornix shortening. But then if you apply a hydrogel at the time of injury, that you can essentially protect the fornix. Okay, we show that kind of in picture format on the far. Oh, you can see my arrow. No. Oh yeah. In this one here, no hydrogel, very bad injury to the fornix. This one is much better, and we show that in H&E and in direct visualization. And we showed through fluorescent imaging. Again, this is in vitro fluorescent imaging in the IVUS technology. Essentially, you put the animal into a box that you can follow the time course of the fluorescence. And we showed that indeed the hydrogel stays there between 6 and 12 hours, as shown there, the fluorescence or lack of it. So you can imagine when patients or potentially soldiers, if we were collaborating with the DoD, which we're working on now, that at wartime, anything that can cause injury to the eye, quickly spurt on a protective agent. Or if you know you're going to have chemotherapy, other injury to the eye, sometimes you know that in advance, or immediately after a plumber is working with Drano, we need to immediately put in the hydrogel and protect that ocular surface, and it'll stay there. Of course, there are other methodologies. This one could be a very good one if it's a topical treatment every 12 hours. Okay. And we showed other molecular markers of fibrosis and that indeed that it can protect. One of the things that we did not expect, but nonetheless helped the fornix from not contracting, was that goblet cells, which are critical for the production of mucin, are not protected by the hydrogel, but nonetheless, it does stop symblephra formation. So the hydrogel is not perfect. There's still killing of the goblet cells. Will they come back? We don't know. So we haven't followed it long enough, but we suspect that they will, because we see a little bit after the hydrogel on the bottom right. Okay, so we created a symblephra mouse model, never done before. It had been done a little bit in rabbit, but rabbits are quite expensive. They do recapitulate the human ocular surface a little bit better. They're bigger, they're easier to work with. But if you want to do replicates, if you want to do large scale testing, mouse is really the way to go. Okay, let's move on to another area. Now, we were very excited about this paper that came out in Stem Cell Reports because it pioneered a couple of technologies. One, single cell transcriptomics, which I'll talk about, and the creation of corneal organoids. Now, organoids is really an outshoot of that initial work that you could create and differentiate the initial induced pluripotent cells. So to go back a little bit, I think I have a few slides. Actually, I'll show that in a moment. So we know the cornea, beautiful, beautiful structure. It's incredibly important for refracting the light, the surface of the ocular surface, the main refractive index, even more important than the lens. Okay, so if you don't have a good ocular surface, you're in big trouble. Okay, we know that corneal blindness worldwide is a major leading cause of blindness, and Fuchs dystrophy, especially in the course of aging. I'll talk about that a little bit more. But by and large, corneal blindness, multiple reasons why the cornea could be injured, and why we might need more tissue. Okay, so go back 5 or 8 years ago. We were creating induced pluripotent stem cells. So I think I will have to chat about that. You can take skin or blood. These are somatic cells, completely differentiated from when you were an embryo to the moment of conception, all the way to the baby is born and to later in life, 50, 60, 70, 80, and beyond. But you can take a skin cell or a blood cell from an adult and express 4 to 5 genes, but largely 4. And the Nobel Prize was given because if you express those 4 genes in the course of about 4 or 5 days, that cell will go back and become like an embryonic stem cell. But it is your own cell, so it's indistinguishable from yourself. Well, what they showed over the course over a number of years that you could differentiate this tissue. You could nudge it not back into a skin cell or a blood cell, but into an eye cell. And that was published in a number of papers, probably around 2014, 2016, but all in 2-dimension. And this is one of the protocols that they had a clinical trial in Japan, looking at, can you create corneal progenitor cells using this technology? And it's really amazing because you have colonies of cells that are like embryonic stem cells. But if you look at the different zones, the central zone, number 1 on the bottom right, first zone, is very much neural tissue. And then if you go to zone 2, 3, and 4, you get neural crest, limbal epithelia, ocular surface epithelial, and surface ectoderm. It really is like a mini eye, but in 2-dimension. And that is really what spurred people to think, well, what if we didn't culture it on tissue culture plastic where it grows out, but we cultured it in media, and ultimately we were able to create. We did not create this the first in the world. There were other groups that did this, but we thought, hey, let's harness this new technology for what we're going to do and what I'm going to show you. So we can create, in our lab, we've created, bottom right, these are our organoids, little mini corneas in a dish, okay? It is painstaking work. It takes 3 months of nurturing these cells from a culture into little spheres that are essentially like little eyes. Okay, the cornea on the back part is neural tissue. So you can imagine if you can create a patient-derived mini eye, what would you do with that? Okay, well, we have a lot of ideas. I'll mention a few things. This is just one of what we did. We thought, okay, if the differentiation, essentially like in the womb, it takes 9 months to go from an embryo to a little baby. Well, largely it takes 3 months in a dish to go from a bunch of little cells into a mini eye. Well, that time course doesn't match. Does the 8 months of development match the 3 months of differentiation in a dish? So how did we approach that? How did we go about looking into that? Well, we said, okay, let's get some fetal tissue. Okay, now it's ethically charged. These are tissues we received from another laboratory that were already using the tissue for the brain. And we were able to get to the eyes. So, okay, we will use that tissue. It's already there. Of course, this has all been approved by RB and our protocols at Stanford. So we did single cell RNA sequencing, which I will have another slide on, but we essentially compared adult corneas with fetal corneas with the organoids. And we can see here that the corneal organoids are shown from taking primary cells, reprogramming them into iPS, and then creating little mini organoids, and then we characterize them. And then ultimately what we hope to do is to take the corneal organoids and transplant them in a limbal stem cell deficiency model. I've already showed you our symblephora model. It's very much like that. If you can injure the cornea and injure the fornix, you can also destroy the limbal stem cells and revive them. So the protocol is about 2 to 3 months, but largely it's actually 3 months to get them fully mature. And you can see them as they slowly become mini corneas in a dish. What do we do with the organoids? Well, we disassociate them into individual single cells, shown in the bottom left. It's a enzymatic digestion. Anytime you digest and you manipulate cells, you will lose some of them. So it's really, we are collecting, it's kind of sample bias of the hardy cells that live. And then essentially in a reaction where there's emulsion of reverse RNA transcriptase that takes the RNA and turns it into cDNA, and then there's barcoding. Every individual cell is labeled and sequenced. That's kind of the bottom line. So you take a tissue, you create single cells, you barcode each cell, and then you do full transcriptomic analysis. So this is only RNA analysis, not DNA. So it's not genomic sequencing, but it's transcriptomic. And you get an analysis of all the cells and what is their transcriptomic profile. The top area shows the cornea in H&E. And what we showed over the course of each month is we interrogated that tissue, and we looked at the expression of every cell. And from looking at the expression of the cell, we could identify what its identity was. So we took a ball of cells and we said, okay, that's an epithelial cell because it's expressing K3 or K12, others that express neural markers, et cetera. And that's how we created that diagram on the left. I apologize for the size of it, but suffice it to say that the list is all the different cell types, and the colors of all the different cells in that tissue, in the organoid, or the fetal eyelid, or the fetal cornea, or the adult cornea. We looked at month 2, month 3, month 4, and we compared it to the fetal and the adult. And we did staining in the actual tissues to show, indeed, we have the tissues that we want, the cells that we want, and what are their locations. And we did that comparison, all the months together, so all the organoids, and we compared individual months as well. In the bottom left, that's one of the figures in our paper showing at month 1, we have a lot of progenitor cells, pluripotent cells, but starting in month 2, that you have more keratocytes. So largely in the corneal organoid, you don't really have cornea formation or the keratocyte until month 2, and then beyond, you're almost completely done by that, month 3 and month 4. So that's the green bottom area. Okay, so in conclusion, organoids are an important tool for transplantation and drug development. This is just the springboard onto more studies that we can do. And we can look at development because we were able to control the moment that it starts to be differentiated till its final product. So I talked a little bit about transcriptomics, and it is a very powerful technology. We were one of the first labs to do transcriptomics in the eye. And this is, again, where kind of being opportunistic and opportunity approaching and being at your doorstep, do you walk through that door? So when I arrived at Stanford, it was just at the beginning of the transcriptomic revolution, I can call it. And there were groups already funded by the Chan Zuckerberg Initiative trying to identify every cell in the human body. And as you could say, if you take the liver and you digest it and you get individual cells, you take the bones. And I talked to them and I said, oh, wow, this is an amazing project. You know, this is a new technology. It's very, very expensive. And the Chan Zuckerberg Initiative wants to fund essentially like the Human Genome Project, but to identify every cell in the human body. But you're not doing the eye? That's a very important organ. Can we do it? And they said, sure. So they would take donors. These were generally healthy donors who had a car accident, motorcycle accident, and whatever part of their body was not okay, we would harvest all the other organs. But by and large, we were looking for donors, young, healthy, not necessarily young, but healthy, and we would harvest every organ in the body. So I'd sent my team, I was there myself, and they would clamp the aorta, and the patient would be pronounced dead, and then there'd be 18 teams waiting to collect every organ. And we were actually, I think, number 2. There were other organs that I thought, okay, we need to get it immediate, but we wanted the retina. So the retina you need to collect very, very quickly. So I think within 20 minutes, we were able to remove the eyeball. And I thought, oh, we might as well get the orbit. Might as well get the lacrimal gland. Might as well get the eyelid. Eyelid was very difficult to get because these patients were also sometimes open casket, et cetera. If you collect the entire eyelid, the family often didn't want that, understandably. But occasionally we were able to get the eyelid as well, and I'll show that work. But over the course of a couple of years, starting around 2017, We were one of these many groups that published this paper in Science as a consortium called the Tabula Sapiens Consortium, and we're very proud to be part of it. One of the first single-cell descriptions of the eye. Okay, but what did we do with that? We were very happy with that. What did we do with that? Well, that again created a baseline. If we can identify every cell in the human eye, we had the tissue for orbit. We had the tissue for eyelid, and I'll show you a little bit of how we proceeded after that. And again, showing that we did the eye, our contribution. So this slide was shown in Journal Club yesterday. Okay, it's a great schematic from Terry Smith, and it just shows a thyroid eye disease. Again, I said we took human orbital tissue. It's very hard to get good human orbital tissue in healthy adult specimen, in a healthy adult. We can get it if we do a decompression, and that's what we compared it to, our thyroid eye disease fat. We can get it during an enucleation, but often those eyes have been damaged. So how can you get a pristine human orbit? Usually through donation, but again, a little bit tricky. So we were able to get normal human fat. We compared it to thyroid eye disease fat. I just skipped that one. We went over it yesterday for most of you. It shows the pathways and molecular pathogenesis theories, but a lot of it's been proven, but not all of it, about why an orbital fibroblast will become activated and expand through the deposition of hyaluronic acid and GAGs, causing the expansion of the orbit, dysthyroid orbitopathy, and optic neuropathy, eyelid retraction, etc. Okay. This is one of the specimens we took. Okay. We did essentially a lid-sparing exoneration, and we were able to get plentiful orbital tissue, muscle, etc. So an exoneration specimen. We went through the whole pathway, which I talked about already. Once you get the data, you go through a long kind of computational analysis, and you get our atlasing. Okay. We had initially 3. This is we actually are adding on a few more patients. We have a number of thyroid eye disease patients. We have a number of controlled patients that we did this initial comparison, which I'm going to show you. This is being put together for publication. So I'll show you most of the data, but there are a few things that we've left out, some of the key genes that we're very excited about. You can imagine if you have a new gene for thyroid eye disease, we are waiting for publication. Okay, but by and large, we've shown almost all of it. We have human orbital fat transcriptomic profiling, a whole array of different cell types, again, to show that we have fibroblasts, we have neural cells, we have dendritic cells, etc. Again, you're able to parse out what are the cells of that tissue. And you can show through conserved markers. Like I'm showing one of the clusters, fibroblast, is that pink cluster on the top left that has all the hallmarks of fibroblast tissue. So that's how we identified it. It's kind of like if you were the first person on a new continent, you look at a tree, oh, it has green, it's wintertime, oh, it's probably an evergreen. So I'm going to put in the evergreen category of tree. Oh, that one has no leaves, or it has big leaves, whatnot. That's how we largely did it. We're walking into a whole forest of cells. How do you identify them? And you do it by the gene expression. Okay. And then you look at the top differentially expressed genes. So again, this is the comparison between thyroid eye and normal orbital tissue. What are the genes that are upregulated in thyroid eye disease? Literally, what is different? And that will give you a hint of what is causing thyroid eye disease. Just that in itself, right, the idea that you can compare the gene expression, every gene that's expressed in that cell at that moment, will allow you to look into almost any human disease process, including aging, which I'll show you at the end. But if you think about glaucoma, people are looking at optic neuropathies, people are looking at essentially every field. This technology is very, very important. It is difficult to do. So that's why there's some expertise involved to get viable single cells and then the analysis. So you kind of need to get the right team together. And again, this is showing our normal and thyroid eye disease transcriptomic analysis. Each number is really a type of cell. So the spatial representation is showing how different the cells are. So it's kind of like if you have a palm tree, it'd be way, way over here. You have an evergreen, it'd be kind of over here. And if looking at different markers, these are just highlights of within all the cells. Each dot is a cell. which cells are expressing adipogenesis genes, mature adipocyte genes, differentiation into adipocytes, and allows you to do that type of analysis. You can look at genes for white adipose tissue versus brown tissue, and they will light up different cells, again, showing that you have different cell types. And one question that people always ask me, what percent of the thyroid eye disease cells actually are expressing IGF-1? And it turns out we could come up with a number now. 45% of the thyroid eye disease cells are expressing highly, more so than the normal human fat, IGF-1. What does that mean? Well, in the US, we're treating with teprotumumab. Well, maybe teprotumumab is only affecting 45 of those cells. So maybe that's why we're only getting 2 millimeters of proptosis reduction and not 4. And why this really gets into why things are happening at the molecular level. Okay. I will keep moving. And we can look at the immune clusters. So we looked at what immune cells are going on in the orbit during thyroid eye disease. And we saw, for example, CD1C, B cell activation. We know what subtype. We looked at mast cells and FcR1. PK1B is a protein kinase inhibitor gene that is important for generalized immune inflammation, T cell activation, neutrophil activation, T cell and dendritic cell activation, and lipid antigen presentation, respectively, on that bottom right panel. You can do RNA velocity, looking at what cells are turning into what cells. So there are different kind of very cool types of analyses you can do. And then you can start to say, okay, where are we in terms of adipocyte differentiation, and where can we impact our understanding into the different inflammatory pathways? Okay, so again, this is very high level. I'm not diving into the details, but just to give you a taste of what we're working on. And actually, I'll just skip that for time. Well, what did we do in addition? Well, I mentioned we collected human eyelid. Well, we just submitted this paper actually 2 days ago, so I'm going to present a lot of the data. And Margaret Chen on the top left is a UofT undergrad. So she worked remotely, a lot of the computational work together with my team at Stanford. Obviously, you need people in the lab. And it's a group effort with a number of key people who are doing this type of work on the bottom right, Irv Weissman and Steve Quake, who's now the head of the Chance Zuckerberg Initiative. So in a nutshell, it also allows us to look at eyelid development. Oh, okay. So eyelid development, we were able to also get fetal eyelid. So we were able to, well, at the same time of getting the fetal cornea, we got fetal eyelid, and we compared adult eyelid with fetal eyelid in single cell sequencing. Well, why would we want to do that? Well, here's the kind of common thinking that over the course of development from embryonic state to full term, that the eyelids develop and at some point will separate. This is largely done through anatomic analysis. Well, now we were able to interrogate it using more of a molecular approach. We did this at the single cell level using the same technology. We were able to get 3 fetal samples at the ages of 20, 19, and 22 weeks, and we compared them to 3 adult samples between the age of 54 and 56. And again, the question is, do we know for sure every cell type of the eyelid? Largely, we follow anatomy. We cut the eyelid, we do a sagittal section, we look at our atlases, and we see, okay, we have conjunctiva, we have tarsal plate. We know within them they're embedded the meibomian glands. We have our sebaceous glands. Up in the fornix area, we have the accessory lacrimal glands. We have Mueller's muscle levator. We talked about that last night as well in Journal Club. But do we know for sure that there are not 2 types of muscle in Mueller's? Maybe. What if the aponeurosis is actually composed of something else that we don't know? All we know is looking at a picture of H&E for the most part, and of course, functional correlate. So we took the full cross-section of the eyelid and we ran it through single cell analysis. We took a little tissue and we did H&E, again, to show all the cell types that we have shown here. And it's beautiful. The eyelid's beautiful, just like the cornea. We have our meibomian glands, the way we have orbicularis oculi, we have conjunctiva. And we largely showed that in our single cell. I apologize for the small picture on the right. This is one figure from our paper, but I blew up the key one, which is over here, which is, again, we were able to show that every cell type largely that we thought would be there, we were able to identify fibroblasts, conjunctival epithelial, satellite cells, Mueller's muscle. Again, there's not much smooth muscle in the eyelid. We talked about that again last night. So looking at smooth muscle markers, we were able to show, okay, we have a Mueller cell population. And I was very, very excited about the presentation because we talked about ocular pharyngeal muscular dystrophy, a genetic mutation. Well, I'm going to frankly talk to the people in Quebec, and of course, if others have samples, we can now do single cell on those patients and show a change in that little population, number 8 of Mueller's muscle. And if we show a change in description of the genes in that population, we pinpointed what are the Mueller's muscle pathology that's going on at a genetic level. Okay, so you can imagine any genetic disease. If you do the comparison, you can show which cell types are involved. And many genetic diseases, we don't know what cell types are involved, especially with the anatomical and developmental changes. Okay, so PAX6 and other developmental genes, we can interrogate it and say, oh, actually, even though this gene is affected, these 5 cell types are the ones that cause the problem. And we can target those 5 cell types individually. So it really allows for a kind of personalized and very surgical approach at getting at a disease process. Okay, same thing for the adult eyelid. We were able to show the cell types. And again, this is the same type of differential gene analysis. What are the genes that are upregulated? And the bottom right, oh, actually, this is fetal. I think this is fetal. So the first one was adult. I apologize. This one's fetal. And these are the individual, was it adult? Yeah, the individual patients. Okay. We did also corroboration with immunostaining, because anytime you take a tissue and you look at the genes, people are going to say, well, is it actually there? You showed upregulation of a gene. Well, we actually took the tissue, we sliced it, we took an antibody to the gene of interest, and we showed it binding. So it's largely accepted proof that indeed the upregulation is what we described. And this is the different eyelid markers, including mucin, goblet cells, conjunctival cells, etc. And then finally, this is the merge fetal and adult. This is comparison of fetal and adult. The blue is fetal, the red is adult. And we could see over the fetal eyelid and the adult eyelid, we have a different population of cells. So early on, there are quite a few fibroblast cells, much more so in the fetal, developing hair follicle, etc. Again, I apologize for the small font, but again, it just shows that there's a whole range of things that we are interrogating, different cell types that would require many, many slides to show each individually. So with RT-qPCR, you're looking at one gene, you're looking at changes in one gene. Here we can look at all the genes at once. So it's very, very powerful. Okay, so we identified and analyzed different eyelid cell types. We confirmed the time frame of eyelid development. I didn't go into details, but we showed that indeed it's about 18 or 20 weeks where there's eyelid splitting because we had that time frame. And it really sets the foundation for future comparative studies. Okay, good. I think the timing is just about right. I'll go 5 minutes. This is probably, I saved the best for last because I'm very, very excited about this. I mentioned about 10 years ago, they came out with the technology to turn back the clock all the way. Well, a few years ago, there was a paper that came out from David Sinclair's group at Mass Pioneer that they were able to turn back the clock on retinal ganglion cells and largely improve potentially vision from aging changes. Well, at the time, our expertise was in the front of the eye. So we thought and we said, okay, well, what if we turn back the clock, not all the way, that same cocktail, which is the expression of 5 genes, using an mRNA approach. So it's an mRNA-directed therapy. Again, you could use other virus or incorporative genes, but that may cause potential malignancy down the road. So it's largely thought if you just put in mRNA, it's generally safe. The mRNA causes, over the course of a week, an adult cell to become an embryonic cell, and it loses its identity. A epithelial cell or a blood cell or whatnot will turn into into something that is indistinguishable from an embryonic stem cell. Well, what if you do that treatment, but for a much shorter time? And that's what we did. And that's called partial reprogramming or transient reprogramming. And the idea, this is really what we're talking about, is right now in normal development, you have fertilization, a creation of embryo, and then over the course of 9 months, you have the creation of an offspring. This is my eldest son, Felix. Okay, so we can create life. And iPSC technology, you can do that. You can take an adult cell, you can turn back the clock and make it embryonic, and they've created an iPS mouse. So you can essentially do cloning. It's very, very scary, but you can do it, okay? With obviously with ethical considerations, which I'm not addressing today. But now with epigenetic reprogramming to go against aging, you can take an aged somatic cell and you can turn back the clock and you can make it young again. Well, what is aging? I firmly believe that in many ways aging is a disease, okay? Because we can now look and interrogate an old cell and a new cell, we can compare what are the differences. So we live now twice as long as we did 200-300 years ago. And what we've now come up with is indeed epigenetic alterations, mitochondrial dysfunction, stem cell exhaustion, telomeres about 20 years ago, it's all telomeres. No, it's one of many, many things that are involved in aging. Mitochondrial function, it harvests and really negates free radicals, which we know are harmful. So if you have better protection, that will also cause you not to age as much. And we know that many diseases are caused by aging, and our population is aging. Major issues for many countries. So how are we going to deal with many of these aging eyes? We know macular degeneration, critical, aging. Okay? We don't see age-related macular degeneration in infants for the most part. Similar changes perhaps for certain types of disease, but by and large, age-related macular degeneration, cataract, diabetic retinopathy, glaucoma, and then certainly for the ocular surface, dry eye, etc. So we know that epigenetic alterations can affect this aging pathway. And our thought was, can we rejuvenate tissue to largely to fight against age-related eye disease? So we wanted to show this in vitro first. We took corneal rims, as shown in the bottom right, and we plated them. So we created a primary cell culture. So this is not tissue culture from a cell line, but these are actually adult cells from a corneal rim. And we looked at what are the cells that are, what is the expression of particular genes that are involved in aging. Okay, and that's shown here. We looked at inflammation, catabolic keratinization, cellular senescence, mitochondrial dysfunction. And what we were able to show was that the young cells, and those were in patients around 20 to 30, were quite different than the older cells, which we overall gathered around 60 and above, maybe a few in the 50s. And what we showed was that we could partially reprogram these cells, and we could interrogate them for these aging hallmarks. So again, we did a 3-day instead of a kind of a week to 2 week long reprogramming. So only a partial reprogramming. And we have cellular rejuvenation in essence. And I'll show you what we found. So again, this is to show that when you have your cells, you can express the OSKN-L, so that's OCT, SOX2, MYC, a number of genes that NANOG, that we express with mRNA, and we had expression. This is largely to show expression in the cell types and to show that there's no expression in cells that were not transfected. And again, I apologize for the small font, but this shows that, well, it is quite small, isn't it? The green, so this is OCT, so the top left is OCT, 4, SOX, KLN, MYC, LIN28, and NANOG. So this is our mRNA cocktail. And again, showing expression of those particular genes in that cell type, in the cells that we've cultured. And in the bottom right, showing, the bottom left, sorry, is ABCG2 for for limbal stem cells, P63 again for ocular epithelia, and SOX and PAC6 for ocular lineaged cells. This is largely to show that while you can do the partial reprogramming, it doesn't change the cellular identity. So unlike full reprogramming where you lose its identity, you can do partial reprogramming and it will retain its identity. The left is young, the middle is aged, and the green on the far right is the aged that had been treated with the cocktail. We showed that we can alleviate senescence and inflammation markers. So in essence, P16 and P21, they're tumor suppressor genes. They're involved in cell cycle. So again, showing that we can alleviate cell senescence, where cells kind of poop out. Young is the expression of P16 and P21, and the aged cells show a marked increase in those gene expressions. But then when you treat with the cocktail, those same cells react and behave like the younger. So essentially, old is the red, young is the blue, and then green is the rejuvenated, showing the same phenotype as the young. And then finally, IL-6, loricrin, and MMP-1 showing markers for inflammation. The red is older, the blue is young on the far left, and then the green, we're able to rejuvenate that tissue or increase the expression of protective factors like MMP-1. We were able to rescue these tissues for mitochondrial function, as well as looking at superoxide dismutase, TFAM, a mitochondrial marker, again showing that the green, essentially the rejuvenation cocktail, is able to bring the cells much more into the young phenotype than the aged. And actually, I'll just kind of move forward. So in essence, we were also able to do this in TNF alpha. So when we insult the cell with something that would cause inflammation, those cells also behaved and were more robust to inflammatory insult. So in conclusion, I'm sorry I went through so quickly, but for time, we can reverse the hallmarks of aging in ocular primary cells. Again, we only have done this in vitro. And then this could be the future foundation for regenerative therapies. Our hope now is to rejuvenate this tissue and to show functional changes, both in vitro, in our colony forming assays, which we're doing now, and then taking adult tissue, rejuvenating it, and then grafting it into an adult animal model to show that the tissue behaves better. And frankly, if it works, in a patient with dry eye, you can apply a few drops of the mRNA cocktail. We can rejuvenate potentially accessory lacrimal gland or conjunctiva. Cells that might be just pooping out, and we can make them more robust again. So I'd like to thank you all for your time. I know it's really a whirlwind, but gives you a taste of what can be done in the lab if you can identify a problem and identify a technology that can maybe solve the problem. I welcome any comments, emails. I really need to acknowledge my lab, shown on the bottom right. I work very, very closely with Irv Weissman, who identified the hematopoietic stem cell 40 years ago. So we're nested within his lab, and we have our own lab space as well. It really could not have been done with support from NIH, donors, Fight for Sight, now RPB. And for all the trainees and others, please scan this QR code. It's about a 1-minute feedback, and it'll go to Stanford just to give me an idea of how well I'm doing in teaching. But yeah, it's great. For all of our teaching now, we do the code, all the students, trainees will input, and then it allows the faculty and the administration to know how we're doing on teaching. So again, thank you so much for your time.
So there was a question for you from Victoria Leon. She's an oculoplastic specialist in the city. What's the time frame for identifying upregulated gene expression or genes expressed in thyroid orbits to developing clinical therapeutics to target those genes?
Yeah, so thyroid is one area that we're very, very interested in. The time frame is going to be tricky. I think as an oculoplastic surgeon, you know that the timeframes are not well defined. What is active or what is chronic disease? Even that is a little bit in debate. We know that the natural history of the disease 1 year, 2 years? And when do we call it chronic? Is it no activity for 3 months, 6 months? And what actually do you call activity later on? So I think it's going to be tricky to get at that. Largely what we've done and what we plan to do is we want patients that are active. Generally active are those that we know are looking at the Rundle's curve that was shown yesterday in Journal Club, that we can show worsening activity, proptosis, all the CAS score, essentially clinical activity score. And when things get really bad, we know they're active. When we know that they start to plateau, we know that it's somewhat improving. So we know kind of what that up curve looks like and perhaps the plateau. So I think we're fairly confident we can get active tissue and identify what is active in our studies. Chronic, I suspect our initial studies will be done at 2 years and beyond. It's very, very rare to have a patient that is chronic beyond 2 years and at least 6 months of non-activity. And we'll want to compare those tissues to look at what genes are involved for active versus chronic. One of the amazing things that we found in the US is that teprotumumab can be used for chronic. We're not fully understanding why that is the case. This is a perfect technology to interrogate that question, looking at patients that have chronic, patients that are treated with teprotumumab and have done well, often still requiring decompression, and then looking at patients that maybe are treated again or patients that have not done well. So that type of analysis, I think, will really get at the question of what is teprotumumab doing in the 45% of cells that we showed, and what are the cells that are not being affected, and for patients that it doesn't work for example. But yeah, great question. The time frame is a little tricky. Thank you.
Albert, I have a question. First of all, congratulations on a outstanding talk. It looks like we're closing in on the fountain of youth. You know, in corneal surgery, donor corneal and corneal transplantation, whether it be DMEC, DSEC, or PKP, the donor tissue typically is from older patients, usually 60, 70. And what we're looking at is endothelial cell counts. We're not really looking at the age or the quality of the, we don't have the tools to look at the quality of the individual cells. Is there any application of what you presented today to perhaps taking these tissues, older individuals, yet the cornea stay clear for decades afterwards? Is there any possibility of taking these older cells from donor patients and applying some of the techniques you used to rejuvenate them, and perhaps they could be even better quality transplant material?
So it's wonderful to see you again, Dr. Slomovic. The application for endothelium is really, really big. I actually didn't get into it, and we've cultured endothelium and we've shown rejuvenation of the endothelium. I think as we know, there's questions whether or not they're endothelial stem cells or what is the regenerative ability of the endothelium. That's been debated. And we know that the curve of endothelial cell counts largely go down and down and down. And it's thought potentially that there is no regeneration of endothelium. It makes it almost the perfect tissue to rejuvenate, as you markedly point out. So we are actively working on that. There are other groups that are actively working on that, of whether or not we can take the tissue. We know that the DSECs, DMECs, they work great, but it would be wonderful if we could do that in the lab. Obviously, there's a huge corneal transplant shortage. Something like 1 in 8 patients worldwide are able to get tissue. In the US, and Canada, we're very lucky that there are donors, and it's not as bad as in other places of the world, but it can be very, very bad where patients don't have any tissue. If we could take the patient's own tissue, rejuvenate it, and then re-inject it, and there are many ways that people are thinking about re-injecting it. Do we want to put it on a membrane, float it like a bubble technique? Do we want to put the patient prone and have other methodologies like magnetic beads and whatnot? Yeah, there's a whole range of kind of practical ways of doing it, but the endothelium is, I think, one of the perfect key things that this technology would work for. And just as an aside, that now that you mention it, you can do another thing to the organoids. We have done CRISPR editing of organoids, and we have a paper in progress where we've put in the call 6A2 gene to essentially create Fuchs dystrophy in a corneal organoid model. So you can create organoid models of human disease and show kind of what is the pathology, and then obviously for testing of potential therapeutics. But yeah, great question.
Very exciting work.
Thank you.
There's another question from our audience. From Stefan Ancon, who's one of our clinician scientists in cornea. And he's asking, are you looking at spatial transcriptomics to localize these cellular populations in tissues?
You hit it right on the head. Actually, in June, we're submitting a R01 on spatial transcriptomics. Maybe I shouldn't mention that. But we are looking at a couple of different technologies at that. Obviously, we have a lot of the framework already for non-spatial. And just to let you know, spatial transcriptomics is, can you take what we showed, that whole dot plot, but then put it into the architecture of the tissue? So you know that that red dot, that's an endothelial cell, is actually going to that, well, hopefully in the eye, not in the blood vessel, unless it's the limbus or elsewhere, for cornea. Can you localize the tissue cellular identity with location? There are a couple of different technologies, laser capture microdissection, people have looked at. Right now, there's a 10x technology, which is good, but it doesn't necessarily give cellular resolution. It's actually be able to put one cell next to another cell. The power of that ability, which I hope comes online in the next 5 to 10 years, is that you can potentially identify the location of the limbal stem cell within the niche, within the crypt. If you can identify that, then you can identify the neighboring, maybe transient amplifying cells, the accessory cells that maintain the stem cell. You know, these are cells that live in the body for almost your entire life, and they kind of replicate every year or every few months. We don't know for sure. They create progenitors that are totipotent and continue to heal the cornea and create epithelia centripetally. So yeah, spatial transcriptomics is a big part. We have a new technology looking at, well, I probably shouldn't mention it too much, but we have a new technology where you take little clumps of digested tissue, 50 cell groups, and we're able to barcode them, do single cell transcriptomics, do fluorescent imaging, and identify what is their identity, and then put them back into that initial fluorescent image. So that will allow kind of piecing together the limbal crypt or the limbal niche, kind of chunk by chunk until you get the whole thing. So, but yeah, very, very important and great question.
So based on the 2D map that you showed earlier, I think number 1 out of the 4 was essentially nerve tissue. What work, how have you started to evaluate how to regenerate the nerve? So I feel like that's a big issue in glaucoma. It's a big issue in many different things.
That also is a great question. In many ways, that area of research is maybe more ripe. There are people working on it. Derek Vanderkooy is very interested in differentiation. As you can imagine, if you could recreate even retinal epithelia, would be huge. And many people are working on the retinal epithelial patches and how do you grow out that tissue? Because you don't, retinal transplants, can you put in individual cells? There's a clinical trial that has taken place now in Japan where they've done subretinal injection of iPSC-derived retinal pigment epithelia. Creating the entire retina would be difficult. There's such an array of cells, but if you can even do RPE, we're really working on kind of baby Legos. We're not there yet, but obviously the front of the eye where you have monolayers, not too complicated in many ways other than the niche. But the retina is a tall order. The retinal ganglion cells, I think, are also of incredible interest, and people are working on that now for rejuvenation. The de novo creation of retinal ganglion cells, I think, is a little bit tricky. There are brain organoids. So that's a whole area of research that people are active at Stanford in the world to create brain organoids. And then ultimately what we are interested in doing, we're working on a collaboration now of, can we create the eye organoid and put it next to a brain organoid? Again, these are all developing. And then see what connections form. That's called an assembloid. and understanding the development of how those signals are initially sent and interrogating it that way. And then can we improve it? It has kind of broad development implications as well as therapeutic. But yeah, great question. Great question.
Excellent, it's a really amazing work. So in your endothelium, so in vitro model, so like we're looking at the markers for like, you know, like more younger cells. So did you also look at like, you know, the morphology? So like the shape, so like whether, you know, after the treatment, the cells may be like more in the like hexagon shape and form like better tight junction, and then did you test that like in terms of morphology and functional change for your in vitro model?
I did not show that, but yes, because when you have the cells, you certainly want to image them, and I think the morphology is as important, well, as important, but very, very important, because again, it's a surrogate for function. Ultimately, the question is for the corneal specialist, if we're able to put endothelium back and they look funny or different, but they're still able to detrigest the cornea, maybe you still accomplish the goal. So there's, I think it's very important, and it could be a good signal surrogate that we are getting rejuvenation or better morphology. But ultimately, we're going to do studies looking at function, particularly at aquaporins, et cetera. Can we get the pump working well? But it may be a question of how well the pump is working or just the number of cells. So you can approach it a number of different ways. So we can get a few cells that are really pumping, or can we just flood more functional cells that are working, but are still able to get the cornea clear, which is what ultimately function. Okay.
So for example, for patient that they don't have enough endothelium cells, maybe there's a way to, you know, use limbal stem cell to reprogram them to endothelium cells. And even like for patient with like limbal stem cell deficiency, maybe there's a way to, you know, collect the endothelium cell, reprogram them into iPSC, and then like through the pathway to do to reprogram, then develop into endothelium cells.
Yeah, that's all very, very, so the question is, can we take endothelium or epithelium and go kind of back and forth if we take the limbal? So that was actually one of the initial objectives of my K Award grant, which is if you take different starting materials, can you get a better end material? So I mentioned we take blood, you take skin, and you turn it into eye tissue. Well, what if you take eye tissue, turn back the clock all the way, and then nudge it forward? Does it maintain epigenetic memory? And you get a better yield, so to speak. And we largely, that paper is actually coming out now, but largely we showed, yes, you get a little, there's some epigenetic memory, but usually in the first early passages, which other people have shown, that even though you turn back the clock, there's some vague memory that, hey, I used to be an epithelial cell, only in the early passages. Once you passage a cell over time, that epigenetic memory is lost. It is a little difficult to take a limbal cell. We know that limbal cells, it's surface ectoderm path, while endothelium is thought to be neural crest derived. So if you, the further back you go, kind of the more difficult it is. So largely, we think limbal epithelial will take us to epithelial line. Neural crest derived cells, if we want to do IPS, is the way to go for neural crest to endothelium. Yeah, great question. Thank you.
Well, one last question for you.
Oh, yeah.
Once you have the abnormal gene expression identified in the lab, how long can clinicians or patients expect for therapeutics in the clinic? Like, what's that timeline?
Yeah, again, great question. What is the timeline between gene identification and therapeutic bench to bedside, so to speak? It kind of depends on a couple of different things. It depends on what the gene is. There are a number of genes, for example, teprotumumab. It's already been tested in clinical trial, but for oncology, if we identify a target that's already been vetted, but a drug company has kind of thrown it out because it doesn't work, but we know already it's safe, the US FDA is like ready to go, especially if we show it in an animal model and we're able to get the drug. So let's just say you identify a gene, there's already great inhibitors or something that will turn it on, so you can not only turn it off, but turn it on. That is a big part of it. So how do you manipulate the gene of interest? The other question, which is actually very, very tricky, but great for the eye, but also tricky, is delivery. Getting a drug to the surface of the eye is actually not too bad. Getting a drug to the retina is very, very difficult. You have to go, and then do you want to deliver it to the vitreous that then goes on and delivers it? So there's a lot of pharmacokinetics involved in the delivery of the target. So you have to get lucky, I think, if you want it quickly. Fastest, I would say 5 years, because you want to do human tests. Again, does the patient want to be involved in a clinical trial? Okay, 3 years. Do they want to already have it vetted and it's been approved through the clinical trial? 5, 8. Honestly, probably 8 to 10 years. They talk about the kind of the valley of death, is that it takes a lot of money too to do a clinical trial, to do the preclinical studies. But it gives hope. A lot of diseases, there are no treatments. So I love this technology for diseases where there are no treatments before. Wow, we have a new target. Let's look at what's available and get at it that way. Yeah.
Well, thank you. You know, you mentioned earlier that you initially wanted to be a retinal surgeon, and so it's no surprise that he's now trying to do injections for oculoplastics, right? Like, you guys have this forever, Peter. I do want to thank you. You did an incredible job this morning. I thought the Journal Club last night was fantastic. So it's been a privilege for us to host Dr. Wu for the last 2 days. Come back. We'd love to have you again. Thank you.
Thank you very much.
And I guess.